Welcome everyone to this session of the Existential Hope Group, which I am very fortunate to be the steward of. My name is Lou. So in this group, we have given ourselves the ambitious task to create the most beautiful futures we dare imagine, futures of radical abundance and flourishing for all, for life and the biosphere. And we're thinking hard about how to do this and how to not extinct ourselves at the same time. Today, we have the pleasure to host Ed Boyden from MIT for a session on accelerating neuroscience progress and cognitive enhancement. There was a very strong interest in this group to discuss cognitive enhancement, and even stronger enthusiasm, I would say, to have you here as a speaker, Ed. I received a lot of messages of excitement about this session, and also a lot of messages of FOMO from those who were not able to attend, but fortunately this is recorded, so there is no reason to have any FOMO. But this is a way to say thank you so much for joining us. We are very grateful, I think, and very excited to have you here today. For those who don't know him and his work, Ed is a professor of biological engineering and brain and cognitive sciences at the MIT Media Lab and the MIT McGovern Institute. Ed leads the Synthetic Neurobiology Group, which develops tools for analyzing and repairing complex biological systems, such as the brain. And his group applies these tools in a systematic way in order to reveal ground true scientific understandings of biological systems, which in turn reveal radical new approaches for curing diseases and repairing disabilities. These technologies include expansion microscopy and optogenetic tools, which I'm sure we'll discuss the details of, but Ed also co-directs the MIT Center for Neurobiological Engineering, which aims to develop new tools to accelerate neuroscience progress. Meaning that from a technical standpoint, a technological standpoint, your work, Ed, is really at the crossroad between many of Foresight's technical lines of work. So your work is intersects a lot with all areas that Foresight is really invested in accelerating understanding and accelerating for the long-term future, such as cognition, nanotechnology, biotechnology, longevity, in particular. So we'll be discussing these intersections. I will first let you start your presentation, and then we'll get into these intersections and also discuss how these technologies are important for existential risk mitigation and, of course, for the long-term flourishing of our civilization and the long-term future in general. Ed, thank you so much for being here again, and the floor is yours.
Great. So I direct a group at the MIT McGovern Institute, which is devoted to developing tools for looking at and fixing the brain. My long-term dream is that we can simulate thoughts and feelings in a computer so we can understand more about the human condition and maybe even help humanity achieve a more enlightened state. In the meantime, we're very interested in trying to cure all brain diseases, and I hope to tell you a couple stories about that today. But not just how we're doing it, but where these things begin. So I'm going to share my screen here a little bit. Is this coming through, the screen sharing? Okay.
Yes, we can see.
Great.
All good.
So the brain is really complicated. Within a cubic millimeter of your brain, there are 100,000 brain cells connected by 1 billion connections. And each of those cells and each of those connections operates with precisions of 1,000th of a second. And so we really build tools that help people understand the spatial and temporal dimensions and to repair brain computations across these dimensions. My goal, as I mentioned, is really to understand how the brain computes. How do we generate thoughts and feelings? And my hope is that we really can understand what it means to be human and maybe help us understand and overcome some of the limitations that cause so much suffering out there. Now, if we want to really make a simulation of a brain circuit, we need at least 3 kinds of tools. We need to watch the brain in action, we have to control the brain in action, and we have to make a map of the brain. And so today I'll tell you 2 short stories about these tools, one about the spatial mapping, how does the brain look, and one about the temporal control, how do we repair the brain's dynamics. So let's start with space. As I opened with, brain circuits are enormously complicated. Brain cells are enormous things, and yet the connections between brain cells are very tiny. They're nanoscale, right? So how do we make a map of the brain? Well, many people have tried to build tools to map the brain. I think we've all seen pictures like this, brain scans. They're very often featured in the popular press. You see these blobs lighting up in the brain, but each blob contains hundreds of millions, maybe billions of brain cells. And we know that 2 brain cells can be doing very different things. At the other extreme, we have microscopes. Microscopes can see very tiny things, but even they cannot see the nanoscale wiring of the brain, the molecules at the connections between brain cells. So in my group, we often think about doing the opposite of what other people do. And so we started thinking, for 300 years, people have been zooming in with lenses to magnify pictures of the brain. What if we grew the brain physically and made it larger? And so there's a whole field called swallowable polymers or smart gels, where they basically study the active ingredients in baby diapers. Add water to a smart gel or a smart polymer, and it'll swell, absorbing the water, and it can grow 1,000 times in volume in a matter of minutes. So we started wondering, what if you could install one of these smart gels or soluble polymers inside the brain? Weave a dense spiderweb-like mesh of this baby diaper polymer inside brain cells and outside brain cells, around biomolecules and between biomolecules. If you did it just right, maybe you could make the brain larger by adding water. The baby diaper material would swell as it absorbed the water, and it would magnify physically by pushing all the molecules apart the brain. So we thought, well, maybe this could work. Now, in image B is a small piece of the mouse brain. Image C is the same piece of mouse brain. About a day and a half later, we've blown it up by 100 times in volume, about 4.5 times in each direction. Here's a movie that shows you a piece of the brain. We formed the baby diaper polymer earlier, and we add the water right there. And I hope you can see, this is a time lapse of about an hour, shortened to a minute. I hope you can see that this piece of brain is swelling before your very eyes. So of course, this is not a living brain tissue. This is a preserved piece of brain, but nevertheless, even having a map of a static brain is very powerful. The basic concept, therefore, is you take a brain cell like the one on the left and turn it into something like on the right, a constellation of biomolecules hovering in space, their relative organization preserved, but they've all been pulled apart from each other. So to the eye, it looks like you just zoomed in. But what happens physically is that the brain becomes 100 times bigger. How does it work? Well, we had to invent a lot of chemistries, and this cartoon shows a couple of them. So in this cartoon, you can see biomolecules in brown, and in purple are little handles that we attach to all the biomolecules. Then we have to weave that dense spiderweb-like mesh of the baby diaper polymer. How do we do that? Well, with chemistry, of course. We can't just dump the polymer on top. We have to weave the polymer so that it goes inside and outside of the cells, around and between all the biomolecules. And if you do it just right, you can make this happen. And when the polymer encounters a handle, it forms a connection. Finally, we soften the brain by adding chemicals, loosening everything up. And then when we add water, the baby diaper polymer swells. But this time, because of the handles, all those biomolecules are coming along for the ride. So the net impact is we can physically magnify the brain. Here's a piece of the mouse brain, and it looks like it's color-coded because it is. What we've done is we've randomly put different genes from jellyfish or coral that encode for glowing proteins. And so every cell gets a unique color code. Now, the wiring of the brain is too small to see, even with a microscope. And so what we have to do is to physically magnify the brain, like I told you. The top middle shows you what the brain looks like if you zoom in with a microscope. It still looks fuzzy. But if we magnify it by 100 times, you can now see the wiring of the brain. And so here's a little movie of a volume of the brain that we now are starting to be able to image because we've color-coded all the brain cells and we expanded it, and now we can see the fine wiring of the brain. So our hope is that we can really make a map of the brain so detailed that maybe we could try to simulate decisions or emotions. Of course, we can also try to pinpoint where brain diseases begin. After all, over 1 billion people around the world suffer from some kind of brain disease, none of which can be fully cured, and for most of which there aren't any good treatments, frankly. Now, this exemplifies sort of a new model that we have of translational science. Invent a tool that shows people science they've never seen before, and then help people make discoveries with it. And the brain is complicated enough that if we help people use the tool, we often help people in many other parts of biomedicine. I'll just tell you about one example. Something like 7 out of the 10 leading causes of death in a normal year, if you can detect them earlier, you can save more lives. Now, why is it hard to detect a disease early? Well, early in a disease, the body looks normal, right? So many doctors contacted us and asked, can we take a biopsy, like from a cancer patient, infuse it with our baby diaper polymer, and make it big enough that those invisible early changes become big and obvious? And so here's a breast cancer biopsy from an actual human patient. We showed that we could magnify physically these biopsies and help improve the diagnosis of cancer. So this is what I call the invent, discover, design paradigm. We build a tool like expansion microscopy, which allows people to see things they haven't seen before. We help people make discoveries based on the tools, and then we design practical implementations that can get out into the world. That's the story I want to tell you about space, and I want to tell you a short story about time now. So of course, expansion only works on preserved things, right? Not living things. And so, well, how do you deal with the high-speed computations of the brain? Every brain cell generates these 1,000th of a second electrical pulses, and brain cells exchange chemical communications also with a timescale of around 1,000th of a second. So we wondered, could we control those high-speed electrical events? And so the idea we came up with is known as optogenetics, opto for light, and genetics because, as you'll see, we use genes to get the job done. The basic idea is brain cells compute using electricity. Can you put basically little solar panels in the brain cells? Shine light on them, the solar panels convert light to electricity, the electricity controls the brain cells. Then you can bring light into the brain. You know, people have been putting electrodes in the brain for decades. Why not put an optical fiber into the brain? Then the question becomes, how do you make the brain sense light? And it turns out the natural world has solved this problem for us. It turns out all over the tree of life, you can find molecules that convert light into electrical signals. For example, here's a single-celled microbe, a green algae, and it has an organelle called the eye spot that we're about to zoom into, which contains all sorts of light-activated molecules. These light-activated molecules convert light into ionic flux, basically an electrical current like a solar panel would. Shine light like from the sun, and you can then activate these molecules and get an electric current. That's how these microbes navigate around in bodies of water. It helps them navigate so they can photosynthesize better. Now, we just got really lucky. I think a big theme of all of this is luck. But we're engineers, so our job is to be lucky on purpose. You can take this molecule because it's encoded by a tiny gene, and you can take the gene and put it into a gene therapy vector, like a virus. I think we've all heard about RNA vaccines against COVID. Gene therapy is kind of going mainstream. So delivering genes into cells used to be sci-fi, and now it's routine. This is where we got lucky again. Brain cells, like this neuron here, can manufacture these little light-activated proteins, and amazingly, they install them in the right place. And even more amazingly, they're fast enough, strong enough, and safe enough that they can convert light into electrical signals. So shine light on a brain cell with this protein, and that brain cell will start making electrical signals, not unlike the ones in your brain as you hear me say these words. Now, what this means is you can take this molecule and install it in different brain cells, shine light on them, and drive them in patterns of activity. Let me just give you one example. There's all sorts of molecules that we discovered all over the tree of life that make this happen. But I want to show you one example from Li Tsai's group at MIT. She took one of her molecules, used a gene therapy vector to deliver them to Alzheimer's mice. These are mice engineered to get symptoms of human Alzheimer's disease. What she found was that if you drive the brain at a special frequency, 40 times a second, 40 hertz, the brains become healthier. This is Alzheimer's, one of the most incurable diseases of all time. But amazingly, if you drive the brain at this frequency, that brainwave cleans up the brain. Now, optogenetics is difficult to use in people. You might have seen a New York Times article a few weeks ago that one of our molecules finally is in human trials and might actually help people see who are previously blind. But still requires gene therapy in living humans. And so if you think about the story I told you earlier about invent, discover, design, it behooves us to think, can we develop a technique that is non-invasive? No need to put a gene in the brain or an optical fiber in the brain. And Li Wei's team discovered that. It turns out you can drive the same brainwaves with a movie, basically. Show the animals flickering lights and deliver to them clicking sounds, and their brains will become healthier. And so now we're running clinical trials with actual Alzheimer's patients of essentially movies, trying to see if we can heal Alzheimer's with a movie. And so it's early days, but some of the early data you can find on medRxiv or other preprint servers is starting to come out, and it looks like maybe it can actually help people who have Alzheimer's disease. Okay, so I told you two short stories where we invented a tool that lets us do science that nobody has done before. We help people make discoveries with these tools, collaborating with the best scientists and doctors we can find on Earth. And then finally, there are designs that are practical that can help people, whether it's early cancer detection or maybe helping reverse brain aging. But my ultimate dream is to understand what thinking and feeling is. And so I just wanted to end on the same note that I started with. Can we simulate brains in a computer? And so one of the ideas is, well, what if we start with some small brains? The history of biology says, don't start with the human brain, start with small brains, and maybe you can then understand deep things about biology. After all, the first genome to be sequenced was not the human genome, but a small virus. So we started looking at small organisms like worms and fish, which have a small number of brain cells. And our goal is to take these technologies and adapt them for imaging and controlling entire small brains. Our hope is in the coming years that we would be able to simulate such brains in silico. So I'll just end on this note that we really think that there are a couple lessons here. One, of course, is deep tools that show you things nobody has seen before are really important for advancing science. But the second is we often hear this tension between translational work to help people and basic science, which is driven by curiosity. Is there a third path? I would like to think that the third path is driven by technology. Invent a tool that shows people what they haven't been able to see, help people discover things, and then design practical innovations that can help people. I just want to acknowledge this is a very large enterprise involving people all over the world. And I don't think there's enough time, unfortunately, in these short form talks to acknowledge all the people who contributed. But maybe I can just end on this slide. We're always eager to help people solve problems. We've helped thousands and thousands of researchers use our technology to study everything from COVID-19 to Alzheimer's to the fruit fly brain to plant seeds to the microbiome. And yeah, don't hesitate to get in touch if you think we could help. I'm happy to take questions.
Oh, that's wonderful. Thank you so much, Ed, and beautiful presentation and call to collaboration. We are very much with collaboration in this group. So just to start with a little bit a question that's very much in continuation of your talk, but to expand even the limits of this potential of these technologies. I have a bit of a three-part question, which requires some background about what we are doing in this group. The premise for the existence of this group is we live at this very special moment in the history of maybe the universe, but our civilization. There are several far-reaching technologies technological revolutions that are happening simultaneously, which open incredible opportunities to manifest visions of the future that are beautiful beyond imaginations, but also these revolutions are happening very fast, which poses significant adaptation challenges. The most problematic of all being the challenges to humanity's very existence and potential, existential risk. So we both acknowledge the need to remain deeply conscious, careful, thoughtful about the way we develop technologies, and we want to dream really ambitiously. And so my three-part question is, what are the biggest risks you foresee with tools and technologies? How could things go wrong? What are the 5 to 10 years most optimistic provisions for these technologies? And on a larger scale, how can they long-term ensure a better future for life in the biosphere and make us more maybe powerful or creative, or help solve our really big problems like micro challenges?
Yeah, there's a lot of questions. Where do you want to start?
I can go through them one by one. First one would be, what is the biggest risk and how things go wrong with these technologies?
Yeah, well, these technologies are very early stage. They're barely being used with people. As I mentioned, the New York Times had an article about one of our molecules being used to help restore vision in a blind patient. Probably the biggest risk is that these technologies are so tough to deploy into the world. I mean, lots of scientists are using them, but that they might not be used. We help scientists use these tools to make discoveries, but I do think that they have a very interesting set of possibilities in terms of being able to help treat diseases or to help cure people of illnesses. And I think one of the biggest risks is that getting things out of academia and into the clinic is tricky because of the fast timescale that venture investing and other industries outside of the sciences expect returns on investment and so forth. In terms of the technology, assume that was solved, which I think is probably the biggest risk. There's lots of other risks that I think are associated with inappropriate use of technologies, but I think people are starting to build structures to deal with that. Of course, any technique that we want to use on an animal or person has to be approved by an ethics board. We have IRBs that review any human experiment with members of the community before it's permitted to exist. Animal protocols as well before anything's permitted to be done on an animal subject. But last summer, our group hosted a discussion about human augmentation, because augmentation may not be covered by classical medical ethics frameworks. As many of you know, there are companies trying to augment the human brain that are not medical in nature. And so I think there's a need for structures that can address those frameworks. Probably many people around this call know more of this than I do, but many people have approached me wondering if you could simulate the brain. What does that mean for AI? And there are entire groups dedicated to the question of AI safety. Again, you all probably know more about this than I do, so I'm preaching to the choir. I'm probably here to learn from you all, but I'm much more on the biology side, but I do hear a lot of discussion about that. Does that answer your question?
Yeah, totally, and thank you. And we can. That's what I mentioned when I said I'm excited to dig into the intersections of the technologies Foresight is reflecting on, and your work, and AI is definitely part of that. But first, now that we've explored the risks, I would love to know for you, what are the 5 to 10 years most optimistic provisions for the use of the technologies you're working on? Like, what do you really hope to see accomplished and happen?
Well, if we can do early detection of diseases, I mean, that's one of the huge problems of our time. As I mentioned, I hope that expansion can help make early invisible changes into big visible ones. As I mentioned, also the work that Liwei led, which was originating with our technology, but under her leadership, if we can halt or even reverse brain aging, that would be one of a great revolution. So I think in the coming years, as these human trials emerge, the techniques that we have developed over the last 15 years, 20 years, are really starting to show that they can potentially impact everyday human life for the better.
Wonderful. And okay, and now if we take it to the very long-term optimistic future, what is, how do you see this? Let's say, what is your most like ambitious vision in terms of brain enhancement, neuroscience, of what we could accomplish and how it could help really humanity, life, the biosphere, like helping solving our biggest challenges, making us better in a way, or perhaps help us cooperate in better ways so that we do not extinct and we can flourish.
Well, I think if we really understand the human condition, then everything could change, right? I mean, my dream is to go beyond enhancement and to achieve a certain kind of enlightenment. If we understand what the human condition is, what meaning is, what happiness is, might that allow us to transcend even the definition of what augmentation is? The problem with the word augmentation is it's based on our current concept of what we desire, what we want to achieve. But imagine we understood a deeper level of what we want. Maybe we would augment in a completely different direction than we all think we should augment today. And that's why I emphasize enlightenment over augmentation. So that's my dream. If we can understand the human condition, could that allow us to go in a direction that might even be deeper than what we want to do now?
Very interesting. And do you see that like happening already, like understanding the human condition in deeper ways through the work you're doing, or are we still like a bit far from this?
I think full understandings are very far off. You asked me for the far long-term goal. This is my 50-year hope. But it's a very concrete plan. Yeah, I mean, and I think we're on a good track.
I resonate with that plan.
Good.
And great, let's aim for that. Okay, so I'll take a few questions from people who are here. Everyone is allowed to unmute themselves, so I'll call on you, of course. Please post your questions in the chat. Josh McNamee, you had a question. Feel free to unmute yourself if you would like to ask it.
Hey, thanks for the talk. That's super interesting. My question was on for like the brain simulation work. What level are you attempting to simulate, like the individual neurons? Like obviously like full atomic simulation would give you the most details. That would have the most computation cost. So like when you're developing these models, how do you do the trade-offs of like information versus computation cost?
Well, the nice thing about computation is that you don't have to pick one path and stick with it, right? You can change the code tomorrow afternoon and take a different path. So the honest answer is we're going to do whatever it takes to get the job done. I don't know the answer yet because we're still getting the data.
Okay, that makes sense. Yeah, but for like the tool building, so if you're trying to let people see stuff that they've never seen before, don't you have to like kind of come up with some hypothesis, like which level would be the most interesting, like as you write the code?
Say that question again. I didn't quite follow.
But is it like, do you have any hypothesis, like as you write the code, like, oh, we want to make a model at this level for like this information? Or is it basically like all context driven?
I'm not a huge fan of hypotheses. I like to go for what I call ground truth. If you look at history of science, physics used to be very nebulous, planets surround stars and so forth. And there was a moment when quantum mechanics and a bunch of other things hit what is currently ground truth, and suddenly we had lasers and computers and cell phones and the internet, right? And then chemistry was, they used to think of, air, earth, fire, and water, and then there was slow progress to like, oh, that's a metal. And then there was a moment when chemistry hit ground truth, where the periodic table of the elements came along, and now you suddenly can make nylon and Teflon and anything you want, right? So why should biology be any different? I want to hold up for ground truth and not just assume that a certain level is the final standard.
Makes sense. Cool. Thank you.
Yeah. I think it's just hubris for us to think biology is a special science. Science is science.
Awesome. Thank you, Josh and Ed. David and hi, you had a question?
Yeah. So I guess you had mentioned kind of your 5- and 10-year plan and what it is that your 50-year vision is. I wanted to mention 2 specific potential, maybe long future milestones, which are, at what point do you think that we will have the ability to create direct brain-computer interfaces that are high bandwidth that people can kind of use the way that we interact with keyboards or other things to access computers? And at what point do you think we'll be able to simulate or upload brains? And I was just wondering, like, obviously you don't, you're not going to tell us, oh, it's going to be on this date, but do you have maybe a 50% confidence interval for like what time frame we'd be thinking about for when those things could happen?
I don't think much about brain-machine interfaces because I don't understand the brain. So yeah, I mean, I always tell people the hard part of neuroengineering is the neuro part, and it's not so hard to make a bunch of wires and put them in the brain, but it is hard to know what to do with the data and where to put the wires in the brain. The brain's so complicated. So yeah, I mean, brain-machine interfaces are really complicated, and I personally don't feel like I'm ready to work on them. Again, because I'm so obsessed with ground truth. I think we should understand and then solve. If anybody else can beat us to it, they're welcome to try. But again, the history of science, if you look at other sciences, suggests once you get down to the ground truth, that's when the revolutions really begin.
So I guess, how far away do you think we are from ground truth? Is that in 20 years, we're likely to be there. In 50 years, we might be there. In 2 centuries, it could happen. Like, what do you think the kind of general time frame for that might be?
My dream would be if we could simulate some small brains in the coming 5 or 10 years. And if the principles that emerge can then be tested in larger brains, like the human brain, then maybe it's not so far off. But again, this is what I call real science. It's not like a cookbook where we just, like making a pancake, we do the steps and you're guaranteed a result in 30 minutes. And so we might find out that the brain is really, 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 really complicated. And then it might take a longer time. But yeah, I don't know the answer to your question.
Thank you.
Great, other questions?
I have a question. I have a lot of questions, but
go for it.
One in particular about neural interfaces and your kind of skepticism or like maybe just lack. I'm glad to hear the kind of lack of hype about it.
I'm not skeptical of it. I just don't know what to say about them.
Right. So I'd want to ask you something and get your feedback on it about them, which is that it seems to me that at least for input to the brain, we kind of have this ultra high bandwidth, ultra high resolution, parallel optical direct interface to our brain called our eyes. It's like, I don't even know how many gigabits per second it is or how you evaluate it. I know people have tried, and it's like, it's non-invasive. We can pour right in through that method, and the retina is actually arguably part of our brain. So it's like, what more could we possibly make? And what more could we possibly hope to throw in to our brains that's better than that? Now, admittedly, I know we could tweak people's emotions and vestibular senses and all these other things that you can't necessarily do through the eyes directly, although you can partially.
Movies.
Yeah, fair enough. So like I'm saying, okay, so what is the point of the neural interface? Is it for output to a computer? Because we're more limited in that. I don't really know what people are hoping to get, other than this nebulous idea that it's a direct interface to the brain and that's, quote unquote, better.
You have to ask them. I don't work on those.
Okay, fair enough. All right, well, I'll think more about questions that are more germane.
No worries. Like even the Alzheimer's stuff, right? The goal zero Alzheimer's, we're still going through the eyes and the ears, So yeah, non-invasiveness has a lot going for it. Like the whole not having to open up your skull thing. That's really a winner.
Right.
Great. Yes. Other questions? These are great questions.
Yes. I think there is an interesting question by Walt in the chat. Walt, maybe you want to ask it, because we work a lot on generally increasing intelligence in all its aspects, coordination, but also artificial and human ones. Walt, do you want to?
Okay, I'll take a chance. Can you hear me? I guess so.
Perfectly.
Yes. So I feel that one of the biggest the root of the existential risk, self-imposed existential risk, is our own cognitive deficit. That we already cannot handle the technology we have, and there's a lot more coming. So there's a huge danger. So the obvious and really only visible solution is to create a smarter species. And I wonder if there's any work going towards that. That's I posted my essay that I've been floating around for a few months. Perhaps others would care to look at it and see what they think. I mean, you can't do that right now, but I left a link. But anyway, is what's going on in that area? I'll shut up.
Yeah, good question. I don't know of any things that provably make people smarter. I mean, I'm intrigued as this whole 40 hertz oscillation thing. If it can help people with Alzheimer's to have better memory and to have a more normal cognitive existence, could it be used to boost cognition? And that is interesting. I mean, again, the data is what matters, right? All the thinking in the world doesn't replace a small amount of data. But yeah, it's interesting to think about whether some of these tools could be used to augment cognition. But I guess we just have to wait till we see the data until we can say anything positive about it.
Okay.
Other questions? Sure. Yeah, go ahead. Sure.
Yes, definitely. Rudy has a question.
I like how data-driven this guy is. Okay, I guess that's referring to me.
Am I being unmuted?
Oh, now
Yes.
Oh, hello, everybody.
You can unmute yourself freely.
Okay, all right. Well, thank you. Hi, everybody. I apologize for being late to the meeting. And if this was, covered earlier, I apologize. Ironically, I was typing the question in. I think it kind of got covered. Many of us might be on multiple types of cognitive enhancers because we're pretty dismayed that the RAM or the various clock speeds of our brain were not what we'd hoped. Although I could tell some of you on this meeting have much better clock speeds than mine. The point being, I'm on multiple cognitive enhancers. And I was just wondering if there was any data that these, for lack of a better word, actually work and what those might be. And again, if this is not, particularly on point, Edward, you can simply feel free to say nothing works and we'll go from there. But do you happen to know?
I don't know. Yeah. I mean, I guess the question is, are there any data sets which have really compared being on a cognitive enhancing candidate versus not? And what does it look like? I just don't know off the top of my head, but maybe if there's studies out there that have tried this, that would be great to know. Yeah, it's just not my area of expertise.
Got it. Thank you. And also thank you for your willingness to stay in your lane and not speculate when there's no data. I mean, I think all of us have to be circumspect about speculating when we don't have the information. And
well, it's not staying in one's lane. It's just acknowledging the underlying truth of the matter, I think. And it's fine to speculate as long as you say you're speculating. But the problem that I have is that the number of possible states is so large that any speculation, I worry, is unlikely to be true. So let's think about it, right? There are 30,000-ish genes in the human genome. So a brain cell could, in theory, just naively be in one of 2 to the 30,000th power different states, which, if you do the numbers, is more than the number of particles in the observable universe, right? And that's just one brain cell. Now imagine all the brain cells in the brain. So my main worry about speculation is just that the probability that it's correct is statistically extremely small, unless you have data to show that it works. So when we go forth and say, hey, we're trying to cure blindness, we're trying to cure Alzheimer's, we're trying to detect cancer early, we go get the data. And it's not that hard. And if you run out of your own knowledge, you can build collaborations, right? And yeah, so I think it's our responsibility just to go get the data. And if you want to do it and can rally the forces to go get it done, it's not that difficult.
thank you.
Great. Other questions?
Yes, Joy, you just posted a question. Do you want to ask directly?
Sure. Dr. Boyden, given that you're so good at innovating, would you talk a little bit about your process for figuring out which tools to focus on inventing? You clearly have some kind of selection process that has really paid off well. What are you doing that's different than what other researchers are doing?
Great question. I think there's 3 things we do that are very different from other research groups. The first is we always try to go for ground truth. We don't want to settle for an approximation. I think a lot of people hope that an approximation will get the job done quick and dirty, but I switched into neuroscience from engineering 23 years ago. And in those 23 years, we have cured 0 things. So if the approximators can win, that's great, but I don't feel like I need to add my name to lists. I want to go for the ground truth. Secondly, we think backwards. There's so much science that could be applied to the brain, but you really need to think backwards from the problem. People think forwards from the solution. Well, how do you know it's going to work? And so when people ask us, how do you activate the brain with light or expand the brain? Why does this stuff always have impact? Well, it's because we're thinking about the problem and not just innovating for innovation's sake. And then thirdly, we collaborate like crazy. So the last slide had literally like 200 names on it. And one of the things we really try to do is to really bridge different disciplines. Some people like to say they do interdisciplinary science. I don't think that's enough. I think we have to do omnidisciplinary science. And in our group, we have a professional photographer and a neurosurgeon, and it's about as interdisciplinary as things get, and maybe toward the words omni.
Great, thank you, Joy, for the question and Ed for the answer. I have a couple of questions to get into precisely the technologies that Foresight is working on. I would like to talk about longevity and AI, for instance, so separately to start with. From your point of view and knowledge and background, what can you say about longevity, maybe age-related disease prevention and maybe mitigation, and things like also brain preservations, like what do you see as being possible and impossible?
Yeah, so I'm not very good at thinking that way. I like to really focus on the data. I think that's one of the themes here. That's one reason why if we can halt Alzheimer's in its tracks, we could try to apply this to other brain aging conditions. And then it's interesting to think about whether if you could halt brain aging or slow brain aging, could that be generalized to halt or slow other kinds of aging? But the field of aging is full of research that's led nowhere, right? And so I'm very hesitant to judge any given piece of aging as good or bad, because this is not my area of expertise. I really want to get the data. And again, if you're in a science that's not ground truth, like brain science or aging science is, then again, the data is all that matters, right? Yeah, I think that's really what we want to do, is just get the trials done and get the data. What was the other question about?
I want to talk about AI, but I'm afraid to get kind of the same answer. I want to ask you what you think about AI from your understanding of the brain, what you think about possibilities for consciousness, what you think about maybe AI timeline, if anything, and what it would be to have, a very different silicon-based hardware for an intelligence. Unfortunately, I don't think there is data on this.
Well, no, but the beauty of computer science is you don't need data, right? Computer science is one of those fields where thinking is enough, right? But if you're asking about like what brain science is influencing AI, I think part of the problem is we just don't understand the brain very well, right? So the kinds of ideas that, as far as I can tell, are dominant in AI now really owe their debt to neuroscience from the 1940s, 1950s, 1960s. But we don't have a map of any complete brain circuit. And so, in a mammalian brain anyway. And so I think the impact of brain mapping on AI is still to come. As far as silicon goes, silicon seems pretty inefficient to me, right? The human brain runs on what, 15 watts? And, AI, again, you all know far more about AI than I do, but it does seem like AI algorithms require a lot more than 15 watts to run. Sometimes they can require millions of dollars of compute power. So I wonder if silicon really is the future of AI.
What could be the future of AI then?
Well, the big surprises in science often come from nowhere, right? Imagine if we were all here sitting around the Foresight of 1920, right? And everybody's doing vacuum tubes, right? Everybody's like, oh, we can make vacuum tubes into the internet, and vacuum tubes into a phone, and vacuum tubes into a calculator. Meanwhile, a few blocks away, the seeds of the transistor and the microchip are being discovered, right? So it's a little bit fruitless to spend a lot of time trying to think of the vacuum tube internet, right? It would run for 1 second and then probably crash as the vacuum tube self-destruct. So yeah, so I think it's very hard. Again, the really big surprises can come from the middle of nowhere. And that's why I think it's going to happen with all these things we're talking about. I think it's very futile to try to predict any of it. I'd rather invent it than predict it.
Okay, great. And we touched a little bit. Oh, Josh has another question. So I'll give you priority, Josh.
Hey, yeah, you keep mentioning ground truths a lot. Do you mind elaborating on like how that differs from like data? Like, would you consider like the central dogma biology a ground truth?
That's a good question. So the problem, and data could be ground truth if you cannot break it down into further truths, right? You know, for chemistry, if you know about the periodic table of the elements and a few bonds, you can probably make any chemical you want. In physics, if you know quantum mechanics and a few other, useful approximations, then you can design lasers and silicon microchips and build the internet and the cell phone, right? So those are two fields that have hit ground truth. And as I alluded to earlier, you could argue computer science with the Turing machine paradigm also is kind of a ground truth. There's a universal machine and all of them are kind of equivalent. Now, some could be more efficient than others, but the Turing hypothesis is that all computers at the end are similar in their underlying mechanism. So biology, I think of, is not a fundamental science. Biology is built on top of chemistry. And the way that the chemicals interact, though, can be understood with concepts of computer science. So I think of biology as, computer science implemented through chemistry. And that's been a very powerful model in our group. It's probably not completely true because, again, I don't think we've hit ground truth in biology. But in answer to your other question of the central dogma of biology, obviously the details of the central dogma are not completely correct, right? For example, there are retroviruses that can go from RNA to DNA, the opposite direction of the central dogma of biology. So the central dogma says DNA goes to RNA, goes to protein, but now we know that RNA can also go backwards to DNA through a process called reverse transcription, and a piece of RNA can become DNA. So the central dogma has clearly been proven not to be ground truth, but the genetic code is not bad, right? That's why genomics is such a great field, right? If you sequence a genome, that's useful to somebody, right?
And would you not consider chemistry just an emergent property on top of physics too, in the same way biology is on top of chemistry?
It is, but the nice thing about chemistry is that if you look at the energy scales that are achieved on Earth, we're not in the middle of the sun or out there at absolute zero in the middle of interstellar space, then there's a whole set of approximations that work a lot, right, for everyday life. And so it's not completely ground truth, but it's a pretty good approximation. You could say the same about computer science, right? Now people are envisioning all these paradigms for quantum computing. And yes, you could push computer science in really weird ways. But let's face it, in everyday computer science, the Turing, von Neumann, Harvard architecture, et cetera, paradigms, pretty darn good. Maybe they'll be different in 50 years. Ground truth might not be permanent. So the same way people used to think gravity was F equals ma, and now people think of it as manifolds being curved by mass, right, in space and time. So. A bit of a practicality flavor thrown in there, right? Good.
Awesome. We got a lot of questions in the chat. Okay, so I realized that I missed a couple of them. Steve Vetter, I missed your a question you'd asked before about the same frequency working on human brains and mouse brains. Would you like to ask it directly?
Sure. The mouse is at 40 hertz, but any physical system, you change the length scale and fundamental frequencies change. And my guess is that it would be actually surprising if the human brain, the optimal frequency for Alzheimer's or whatever effect you're comparing would be the same. And so I'm just kind of wondering, is anything being done to actually figure out what is the optimal frequency for the human brain as opposed to just assuming it's 40 hertz because it is for the mouse?
Absolutely. I just put in the chat window a link to a paper that I'm part of where we actually measure these in the human brain and look for the resonances. And it's pretty much on cue. I mean, and one reason why maybe brains at different sizes have similar temporal properties is that we're all dealing with the same environment, right? Mm-hmm. If a mouse is trying to escape from a lion and I'm trying to escape from a lion, maybe our brains are dealing with similar things. And also our muscles aren't that different, right? I mean, a mouse is probably a bit faster than me, but maybe not actually. I haven't thought about this. But we're
tennis shoes on, right?
Yeah. So this is something I just put in the link of paper. It says that, hey, 40 hertz might not be so bad for people, but it's a small study. it was hard to do human trials with COVID. But the small study that Li Wei and a bunch of other people, including myself, under her leadership did, suggests that maybe 40 hertz isn't so bad for people in terms of actual Alzheimer's treatments.
Okay, great.
And Rick, you had a question too? Earlier that I missed. Can you identify the activating and suppressing synapses?
So the last time I checked, which is a little stale, people were running models. We had the connectome for C. elegans, but in many cases, we don't know if a synapse is, I'm not sure this is the right term, activating or suppressing.
That's right.
Synapse.
That's
Can
correct.
your technology nail that down?
Exactly. So first of all, the connectome of C. elegans, if I recall, is not an actual connectome, but a mixture of 6 different organisms. And so, it might not add up. We don't know for sure. So our hope is that our expansion method could give us actual full connectomes of individuals. But as you know, you also want to know what molecules are there. Are they activating or suppressing? And that's one of the nice things about our technique is that all those glowing images I showed, that's us imaging molecules. So we can tell you whether a connection is activating or suppressing. In fact, we're doing that right now.
Excellent.
Thank you. Sean, I'm going to give you the floor. You asked a lot of questions. They're all about mostly collaboration. So perhaps you can give us a sort of synthesis of your reflection and questions and ask them all at the same time.
Okay. Yeah, sorry about that. Okay. So what do you find to be the most annoying current barriers to online collaboration? Is one way to think of it. I have a number of questions in this arena that can approach this space.
I guess I haven't really thought about that way. What are the most annoying barriers? I just take it that building collaborations is my job, and it's my job to invent strategies to make it work. So I mean, maybe I could answer a question if you asked it slightly differently. And I see in the chat whether you do ask it differently. So
I've got a few variations. So what are your pet peeves with current collaboration tools?
My collaboration tools are pretty old fashioned. It's a pen and paper and my throat. And I just talk with people. And that's because if you want to build collaborations that work, there are 3 things you need, and I don't see any shortcuts about it. The first is you have to build trust, and there's no way to build trust but to talk to people. The second is that you have to find the willingness to collaborate. If somebody's too busy, or they're ill, or they're leaving their job for a different thing, then they're not going to have the bandwidth to collaborate. And then finally, you have to be useful, right? And so the use is how you have to understand how I would help them and how they would help us. So those are the 3 things I think are important: trust, bandwidth, and use.
Okay. So what are your favorite tools and techniques for working with such large groups of collaborators? Maybe you're kind of answering that already, but.
Yeah, I think it's very old fashioned. I mean, I've collaborated with so many people. I mean, our group might be one of the most, maybe the most collaborative neuroscience group on earth at this point. But yeah, there's no shortcut for just talking to people and gauging those 3 things. Do we trust each other? Are we useful to each other? And do we have the bandwidth to connect in some way?
So what tools are you actually using? Are you using Zoom? Are you using Google's tools? Or what? How does it work? Like, what does it look like to see you engaging with a group like that? That's insane. The number of collaborators you have. It's amazing.
So my job is to be the most accommodating person in the room. So I use whatever tool the other person wants. If I'm talking to a retired expert who's now living off of Alaska and only has a cell phone, I'll just call a cell phone. If I'm talking to a high school student who's energetic and wants to contribute and is only on Skype, we'll use Skype. I can't be picky if I want to be coordinating people.
Okay, here's another one. How about, might science move faster if there was software which directly supported the small steps of collective scientific sense making?
It's a good question. And I thought a lot about this, but I don't have an easy answer for it because I worry so much about the science is the little things like, oh, I thought you meant that by that word. Oh, really? You meant that? And I have not found a way around the messiness of slow, boring interaction. And in fact, it's that often is where the most creative aspects are happening. I mean, somebody asked about consciousness earlier, right? And so I've been organizing a series of discussions on consciousness. And of course, one of the assess whether anything is conscious, right? I mean, you don't know for sure if I'm conscious. Maybe I'm just a really accurate robot and the real Ed Boyden is over on the beach somewhere sipping a margarita or whatever they sip on beaches right now. And so, but you might not know that if somebody's defining consciousness as attention, and a second person is defining it as qualia, and a third person thinking of it from a Buddhist philosophical standpoint, right? So I often find there's no substitute for the messiness of just working your way through the topic.
So what would you want of a dream suite of tools for helping you capture the misunderstandings and disambiguating and cleaning them up? How, like, would it be being able to capture the video streams and then, oh, it's at that moment, that was the moment that we misunderstood one another, or basically being able to pin the video streams, pin the audio streams, deal that, like,
I'm not
what
sure.
would help you?
I'm not sure. I mean, very often after a meeting, we'll do like an email follow-up just to make sure we all got the summary. I'm pretty low tech when it comes to, even though I started as a computer scientist and physicist and electrical engineer. What I found is that in biology and other pre-ground truth sciences, 95%, I'm just making the number up, but let's call it 95% of the struggle is ambiguous. You know, if it's the kind of thing we can codify, we'd be done with it by now. Yeah, I don't have a good answer for you.
Well.
But, you know, when I started my group at MIT 15 years ago, I invited a bunch of like anthropology types and business school types to just look over our group's shoulder and study everything we did. And I said, look, this would be a great opportunity. You can see the birth of modern neurotech. Nobody took me up on it. And I think that was a huge missed opportunity because, you know, looking back 15 years, you know, well, a big chunk of what neuroscientists do now comes from, you know, groups like ours and a few others. And it would have been a great opportunity to actually study not what we think is important, but what actually is important. And yeah, I regret not pushing harder for that because I've been trying to actually write down what I think was important, but is it just my biased memory? Maybe what was actually important was something completely different, and I might be wrong.
Is there an opportunity for that task to be rejoined?
It's possible, yeah. I mean, I think neurotech's going to be very exciting over the next 15 years too. You know, I often thought, what would it be like if you could watch Silicon Valley between the transistor in 1947 all the way to the PC in the late 70s, right? That would be one of the greatest history studies of all time, or chemistry or quantum mechanics in the 1880s and the 1920s respectively. And I think brain technology between, you know, the late 90s when neurotech started undergoing its modern renaissance to today is the beginning of something, but it's going to keep going. It's just the beginning.
Awesome.
Thank you.
That's exciting. And thank you, Sean, for really getting into this discussion. Ed, we hit the hour mark. Do you have time for one more question, perhaps? Or do you have
one, but I am leading a multi-group meeting, speaking of collaboration now, and one of my rules is I don't need too late for that because it sends the wrong message.
I think we should leave it there then, because it was a beautiful way to end also on thinking about collaboration and collaboration tools. This is very much what this group is about.
Wonderful. Glad to hear it.
Yeah, thank you so much for coming here today. This was a super interesting discussion. Lots of thanks and gratefulness in the chat. Really appreciate and wishing everyone a really good night, and see you at the next session for all the Existential Hope group.